I, Dr.Malarkodi, Professor, Department of Pharmacognosy, Vistas, going to give a general introduction about a volatile oil containing drug cinnamon. These are the contents. Introduction, biological source and geographical source of the bark, macroscopy and microscopy of cinnamon, powder microscopy, chemical composition, chemical test, uses and extraction of cinnamon oil. Introduction. The bark of various cinnamon species is one of the most important and popular spices used worldwide, not only for cooking, but also in traditional and modern medicines. Overall, approximately 250 species have been identified among the cinnamon genus, with the trees being scattered all over the world. Cinnamon is mainly used in the aroma and essence industries due to its fragrance, which can be incorporated into different varieties of foodstuffs, perfumes, and medicinal products. The most important constant of cinnamon are cinnamic aldehyde and trans-cinnamaldehyde, which are present in the essential oil, thus contributing to the fragrance and to the various biological activities observed with cinnamon. The characters of cinnamon, synonym, cortex cinnamon, cinnamon bark, dalchini, and Ceylon cinnamon. Biological source, it consists of the dried inner bark of the coppiceous shoots of Cinnamomum xylanicum. Geographical source, the genus contains about 250 to 350 species worldwide, distributed in tropical and subtropical regions of North America, Central America, South America, Southeast Asia, and Australia. Approximately 21 species of this genus have been recognized in the peninsular Malaysia alone. A number of these species are used in traditional medicine and as spices, especially in food, fragrance, fumigants, and in traditional medicines. Macroscopical characters. Color. The outer surface is dull yellowish brown and the inner surface is dark yellowish brown. Odor is highly fragrant. Shape is compound quills. Size of the bark is about 1 meter in length and 1 centimeter in diameter. Taste is aromatic and sweet, followed by warm sensation. Fracture is splintery. Macroscopy of cinnamon. Occasional patches of cork and cork can be seen. So the outermost layer consists of continuous band of pericyclic sclerites with a small group of pericyclic fibers embedded at intervals, and the primary phloem cannot be distinguished. Next layer is the secondary phloem made up of parenchyma containing the oil cell, mucilage cells, and phloem fibers. Sieve tubes are embedded in the phloem parenchyma. The phloem parenchyma is composed of thin-walled cells with yellowish-brown walls and contains starch and simple numerous acicular crystals of calcium oxide, and some phloem parenchyma contains tannins. The oil cell may contain volatile oil, and the mucilage cells contains the mucilage. The phloem fibers occurs in isolated conditions or in tangential rows, and they are more abundant towards the inner part of the bark. The secondary phloem is divided by radial medullary rays, which are uniseriate to biseriate near the cambium, but become broadened outside the tangential growth of the cells. The medullary rays are about 7 to 14 cells in height, and the medullary rays are radially elongated with thin-walled yellowish-brown contents containing the acicular crystals of calcium oxalate. Powder microscopy. The first seen character is the phloem fibers, which are highly lignified, and they consist of definite pits and canals. Next comes the sclerites. The third is the characters of starch granules. The fourth is the cork in the surface view. Fifth character is the phloem parenchyma with the oil cell. Sixth is the part of the medullary ray with some of the cells containing acicular crystals of calcium oxalate, and calcium oxalate contains in the individual cells. And lastly, part of the fiber with an associated oil cell and phloem parenchyma. Composition of the essential oil. The composition of the essential oil of different cinnamon species has been widely investigated, and the oil was found to contain cinnamic aldehyde, linalool, and camphor. Apart from the cinnamic aldehyde, the rest of the constant that is present in the bark is terpene 4-ol, 1,9-cineol, eugenol, safrole, geramicrin D, A-terpinol, A-cadenine, 1,6-octodiene-3-ol, 3,7-dimethyl, and 1-phenyl propanol 2-diol diethonate are the major components seen in the oils. The chemical constants of volatile oils. The principal constant of the volatile oil is about 0.5 to 1%. The volatile oils are present in the unicellular oil cell in the secondary phloem, and the volatile oil contains about 50 to 75% of cinnamic aldehyde and 5 to 10% of eugenol. Apart from this, the drug also consists of terpene hydrocarbons, phelidrine, pinene, caryophyllines, and other terpenes. Further, mucilage, photobotanians, starch grains, calcium oxide crystals, and a sweet principle called mannitol is also present in the drug. The volatile oil, when fresh, is light yellow in color and changes to red on storage. The specific gravity of the oil was found to be 1.00 to 1.030. The optical rotation was found to be 0 to 2 degrees C, and the refractive index of the oil was found to be 1.562 to 1.582. The chemical constants, genuine volatile oils contain about 4 to 10% of phenols, chiefly of eugenol. Phenylalanine is a precursor of both cinnamic aldehyde and eugenol present in the plant. The inferior qualities are generally more mucilaginous and contain volatile oil of inferior fragrance. The drug yields about 4% of ash, 26 to 36% of crude fiber. Chemical test of the oil. 1 drop of volatile oil is dissolved in 5 ml of alcohol, and to it add a drop of ferric chloride solution. A pale green color is produced with the ferric chloride. Cinnamic aldehyde gives brown color, and eugenol, because of the phenolic OH, gives a blue color, and an intermediary green color is produced. Second test is to the powdered drug, add Sudan 3 and view under the microscope. An orange-red oil globule is seen, indicating the presence of the volatile oil. Extraction. Fresh bark sample was washed, air dried, and then dried sample was grinded into powder, and the sample was stored in a drying oven for further analysis. Extraction of cinnamon oil was carried out by means of use hydrodistillation process. Sorted extraction can be also carried with the hexane, dichloromethane, and petroleum ether can be used as the solvents. Extraction of cinnamon oil. The cinnamon essential oil was extracted by means of using hydrodistillation with a Clevenger type of apparatus. For this process, about 30 grams of the precisely weighed of cinnamon bark powder has been put into a 500 ml of distillation flask with 4 boiling chips, and 300 ml of distilled water was poured into the flask. They were immersed for 1 hour, followed by heating at 100 degrees C for 6 hours. Then the distillate was transferred into a 250 ml conical flask, and the volatile compounds are extracted from the water phase 3 times using dichloromethane and a dehydrated over anhydrous sodium sulfate for 30 minutes and filtered through a mid-speed filter paper. The cinnamon oil obtained was concentrated and stored at 4 degrees centigrade for further analysis. Uses. The bark is used as carnative, stomachic, mild astringent, flavoring agent and stimulant, antiseptic, and commercially it is used for losing the weight, used as spice and condiment, and in the preparation of candy, dentifrices, and perfumes. These are the references. Thank you.